Hello students, welcome to the class. Today we'll discuss about the immune system. About the immune system, how immunity is there in our body, forming our immune system, and how immunity can be acquired in our body. Two different types of immunity are there: innate immunity and acquired immunity, non-specific and specific defense mechanism, and which forms the immune system of our body, which is throughout distributed in our body. Immune system, it is present throughout our body in different cells, in different organs, lymphoid organs. Okay, so let's discuss about the immunity, that is innate immunity and acquired immunity. So what exactly is immunity? How can you define immunity? Immunity is the ability of an individual to resist infection even after exposure to a particular effective pathogen with antigenic properties. Means after infection by a particular pathogen, we will not develop the disease. Okay, so this is the resistance of the body. So immunity is the resistance of the body to prevent any of the diseases. And this immunity, how it is developed? This immunity is developed by three different methods. First one is by diseases. See, if we are exposed to any of the pathogen and we are suffering from any particular disease, then by suffering a particular disease for the first time only, the immune system, it recognizes the particular pathogen, and if afterwards we'll infected by the same pathogen, then the immune response will be there. Okay, so by diseases also the immunity is created in our body. And second one is vaccination. Vaccination already we have discussed in the previous classes. By taking the injections or by intaking the attenuated or weak pathogens which do not cause diseases, but they can trigger antibody formation in our body against a specific disease. This is called vaccination. Okay, so vaccination also triggers immunity. Third is by antitoxins. Antitoxins are synthetic antibodies or the antibodies which are produced by different animals, or these are synthetic antibodies and are injected in our body to develop short-term immunity against a particular disease. Okay, so there are three methods how to develop immunity. And what are the types of immunity? The types of immunity, these are two different types: non-specific defense mechanism, that is innate immunity, which is also known as the inborn immunity, and second is the specific defense mechanism or the acquired immunity. This acquired immunity we gain by medication or by vaccination or immunization. Okay, so two different types of immunity are there: acquired is innate immunity and second is acquired immunity. Innate immunity is the inborn immunity which is already there in the body of the individual. So come to the first one, innate immunity. Innate immunity is maintained by four different types of barriers. First one is the physical barrier, second physiological barrier, third is cellular barrier, and fourth one is inflammatory barriers are there in our body to maintain the innate immunity. It is the non-specific immune system, means it is not specific for a particular pathogen, not specific to avoid a particular disease, but it is for all different types of pathogens and to avoid all different types of diseases already there. All these barriers are there which prevent any of the disease causing, which prevent the entry of the pathogens, and even after entry of the pathogens are killed. Okay, so what are the barriers? Let's discuss in a very detail. First one is the physical barrier. Physical barrier, from the name itself, many physical barriers are there in our system. First one is the skin. What about the skin? Skin is covering the all the visceral organs and all the total body throughout our body. And skin, the outermost layer of skin, it is made up of the dead cells, and this layer is called stratum corneum. This layer, it prevents entry of any different types of bacteria or viruses unless any wound is there in the skin. Okay, so skin acts as a physical barrier. Second one, about the hairs. In many places of our body, hairs are present. One of the example I have given here, nasal hairs. The nasal hairs which are present in the nasopharyngeal cavity, here in the nasal cavity, these hairs, they trap the microbes, they trap the dust particles when we inhale the air. They filter the air. Okay, so the nasal hairs, they also act as the physical barrier. Third one is the mucous membrane. Mucous membrane is present in respiratory tract, in the urogenital tract, in our digestive tract also mucous membrane is there, which secrete the mucus. Mucus contains the lysozyme. It is a enzyme having antibacterial property. So the mucous membranes of the body, they also act as the physical barrier. And fourth one is peristalsis. See, peristalsis is what? It is the longitudinal movement of the longitudinal and circulatory movement of the muscles of digestive tract, so that any of the antigen, if it is engulfed through our diet, through our food or water, then it is by peristaltic movement it is removed out. Okay, so peristalsis. And fourth one is tear. Tear, it is the secretion of the lacrimal gland, and this tear, it is also containing the lysozyme, and this tear is also antibacterial in property, and it removes the bacteria from the conjunctiva, that is the outermost layer of eye. Okay, then next is urine. Urine is also having antibacterial property. The urine, it removes the pathogens or any of the bacteria which are there in the urethra. It cleans the urethra. Urine cleans the urethra. And this last one is internal friendly microbes. Many different types of microbes are there which are present on our skin and in the visceral organs, in our different tracts also, in the digestive tract, in the urogenital tract, in the respiratory tract also. Different microbes are there which are friendly microbes, which prevent the entry of other infectious agents. Okay, such as internal friendly microbes, E. coli, it is hugely present in our digestive tract. It produces the vitamin B12 and also kills the infectious pathogens. Okay, so and the urogenital tract also contains the microbes. Supposed to be the vagina in case of female, it contains the different types of friendly microbes which prevent the growth of other different pathogenic microbes. Okay, in the urogenital tract. So all these are the physical barriers. Second come to the physiological barriers which are present inside our body. First is the fever. What exactly is the fever? Fever is not a disease, it is just the immune response. Whenever any antigen it enters into our body, then this is the immune response, the immune system, it responds as the antigenic entry. So it causes fever, or means automatically there will be enhancement of our body temperature, so that multiplication rate of the particular pathogen it is avoided. Okay, and fever also causes, it also triggers phagocytosis of the pathogen by phagocytosis of the pathogen by the WBC, by the phagocytic cells present in our body like macrophages. Okay, and second one is secretions. Different secretions are there. Already we have discussed that mucus, tear, all these are the secretions. And another secretion supposed to be on our skin also. But the sebaceous glands are there which secrete the oil, and sweat glands, they secrete the sweat. These are the secretions which are present on, which are secreted from outside of the body. The sweat and the sebaceous gland secretes the oil. These are also having antiseptic properties, so that it prevents the growth of bacteria or any of the different pathogens on our skin. Similarly, secretions are there from different tracts of our body, so that different types of pathogens can't grow there. Okay, and cerumen. Cerumen is the earwax. Cerumen is produced from the auditory meatus of the ear. External auditory meatus of the ear is the ear opening, and then external auditory meatus is there, which moves and ends with the eardrum. This external auditory meatus, it secretes the cerumen, it is the ear wax. This ear wax, it also prevents entry of the pathogen and growth of different types of microbes. It traps the dust particles also. So it is also one type of physiological barrier, the cerumen. Then next is lysozyme. Already we have discussed that lysozyme is present in our tear, in our saliva, in the lysozyme is also there in the urine, it is also there. Okay, it is having antibacterial property and it kills different bacteria. Then interference. What exactly are interference? These are nothing but the glycoproteins produced by the virus infected cells. These interference, they do not kill the viruses, but they alert the neighboring cells which are not already infected by the viruses. They move from, they are produced by the virus infected cells, these interference, and they move to the neighboring cells, so that the other cells will be will be alerted, and there will be no further growth of viral particle or viruses inside those cells. Okay, so it prevents growth of viruses, it prevents entry of viruses and infection of viruses to the new cells. Next is Hcl of stomach. See, the hydrochloric acid which is produced from the stomach, this gastric gland, it produces HCl, and this HCl, it is having the pH of 2, it is highly acidic in nature. So any of the microbes which enter into our stomach through our food or water, these are already killed in the stomach by the action of HCl. And next is human milk. Human milk, it is having antibacterial property, and it is secreting the lactoferritin and neuronic acid, which are also preventing the growth of bacteria and any different types of pathogenic entry. It also prevents, and it is also antibiotic by nature. Okay, then last one is semen. Semen is the sperm containing fluid which is released from the males during the sexual intercourse. This semen, it contains the zinc, and the sperms, the male gametes, they also cause phagocytosis of the pathogens, any of the bacteria. Okay, so semen is also having antibacterial property. Then come to the third one, cellular barriers. Different cells are there in our body, in our blood. WBC is the number one but fighter cells of our body, and many different types of modification of WBC are also there which act as phagocytic cells, and many different examples I will describe here. WBC, see, WBC is the white blood corpuscles or the leukocytes. Whenever there is any infection in our body, then the WBC count automatically increase, so that the WBC number increases and there will be more and more phagocytosis of the particular antigen. And this increase in number of WBC during any infection, it is called leukocytosis. These cellular barriers, they maintain the second line of defense. See, physical and physiological barriers, they maintain the first line of defense. Okay, WBC, there is an increased number of WBCs, leukocytosis. Next, neutrophils. Neutrophils are the granulocytes, or the granulocytes and the agranulocytes. So different types of WBC are there. Already we have discussed in the previous classes where I have taught about the blood. And these neutrophils, they cause amoeboid movement. They can move throughout the capillaries. Okay, they cause amoeboid movement and also trigger phagocytosis, and they produce the antibacterial phagocytonin and lactoferritin. These two, phagocytonin and lactoferritin, they also increase the phagocytosis rate. Okay, so neutrophils are phagocytic by nature, and they can move throughout the capillaries, finding out the particular antigens or pathogens present, and they kill them by cell eating process, that is phagocytosis. Next, come to the basophils. Basophils, they act as anticoagulating factor, and they also destroy the antigens. Then the monocytes. Monocytes and lymphocytes, they come under the agranulocytes of WBC or leukocytes. These monocytes, these are also phagocytic by nature, they cause phagocytosis, and these monocytes, they can also move out of the capillaries, from the thin walled capillaries into the tissues. This process is called diapediasis, and they can act as macrophages. They enlarge in size and they can act as macrophages. The monocytes develop into macrophages. Next, about the lymphocytes. Lymphocytes are also agranulocytes of WBC. Two different types of lymphocytes are there, B and T. Okay, B lymphocytes, these are processed and formed in the bone marrow. T lymphocytes are produced in the bone marrow, but these are processed or matured in the thymus. Already we have discussed all these things in the previous classes. The B lymphocytes, they maintain the humoral immunity, that is blood mediated immunity, and T lymphocytes, they maintain the cell mediated immunity. We'll discuss later in a very detail. Then come to the macrophages. Macrophages, just I have told you that monocytes, they cause diapediasis, and they enter into the tissue level and increase in size, and they can move throughout the body by the amoeboid movement also. Okay, so macrophages, they cause phagocytosis, and these macrophages, they are having large number of lysosomes, so that any of the antigen, any of the pathogen which is engulfed by these macrophages will be easily digested. Okay, and there are two different types of macrophages. Wandering macrophages, which move throughout the body by amoeboid movement and detect the pathogens and cause phagocytosis. And second one is fixed macrophages, which are present in a particular place in the lymphoid organs. These fixed macrophages are mostly found in the lymph nodes, spleen, it is also one of the lymphoid organ, liver and bone marrow. Okay, so this is all about what? Different cellular barriers. Then come to the antimicrobial substances, which are produced by different cells also. It also comes under the cellular barrier. These antimicrobial substances include biochemical tissue constituents, interferons, complementary proteins. Okay. Then see, first one is biochemical tissue constituents. These are nothing but the polypeptides. Biochemical tissue constituents are the polypeptides which are produced by different cells just to avoid the entry of the pathogen and to growth of the pathogen inside our body, and they can also kill the pathogens, these polypeptides. For example, Bacillus anthracis is causes the anthrax, and this type of bacterial growth, it is avoided by the secretion of polypeptide, that is a biochemical tissue constituent, okay, inside our body. And second one, interferons, already we have discussed interferons. See, these interferons are produced by the viral infected cells, and they prevent more infection of viruses, and they alert the neighboring cells, these interferons. Okay. Then third one is complementary protein. This is much more important, that complementary proteins, there are about 20 different types of complementary proteins secreted, produced by our body for self-defense. These complementary proteins, these are activated by antigen-antibody complex formation. Whenever any antigen is injected into our body and entered into our body, then there will be antigen-antibody complex formation. Antibodies are the self-defense proteins which bind to the antigen, forming antigen-antibody complexes. So whenever any antigen-antibody complex is formed, or any chemical marker of the microbes, it is the on the skin of the on the cell wall of the bacteria, on the cell wall of the pathogens, chemical markers are also there, which are known as chemical markers of the microbes. These antigen-antibody complex or the chemical markers of the microbes, they stimulate the production of these complementary proteins. Okay, complementary proteins are stimulated. Then these complementary proteins, they rupture the cell wall of the microbes by porphyrin production. Porphyrin is a particular type of protein which causes perforation or pore formation in the wall of the microbes, in the wall of the pathogens, by killing them. Okay, and these complementary proteins, they also stimulate the mast cells. Mast cells are present in our connective tissue. These mast cells, these are stimulated by the complementary proteins to release the histamine. Histamine, it is a vasodilator. It causes vasodilation. From the name itself, vasodilation, this term indicates that vaso means vessels, blood vessels. Dilation means increase in width. Okay, histamine causes vasodilation so that the width of the blood vessel will be increased, and more amount of the monocytes, more and more amount of WBC, the phagocytic cells can enter, and they can they cause phagocytosis of the particular antigen present in a particular area. Okay, so the mast cell, it secretes the histamine which causes vasodilation so that more and more phagocytosis occurs. Then these complementary proteins, they also cause complement fixation. What is complement fixation? These complementary proteins, they bind to the antigen-antibody complex and they remove it from the tissue level. Okay, that is called complement fixation. And next is opsonization. These complementary proteins also cause opsonization. What is it? The antigens, the pathogens, these are coated by the complementary proteins. They secrete a particular type of chemical so that there will be a coating formation surrounding the pathogen, surrounding the antigens, so that there will be easy engulfment process, easy phagocytosis process. Okay, so this is called opsonization, formation of coating surrounding the pathogen's body so that there will be easy phagocytosis. So all these are the properties of the complementary proteins, and they maintain the defense system. Okay, next, the natural killer cells. These natural killer cells are present throughout the body, and they also can move throughout the body by amoeboid movement, and they kill the pathogens by phagocytosis. Okay, and the fourth one is inflammatory barrier. Already we have discussed inflammatory barrier is there here. See, the mast cells, they are present throughout the connective tissue, and connective tissue is distributed throughout the body, so mast cells are present everywhere, and these mast cells, they secrete histamine when any of the antigen entry is there in our body, into the cell, into the blood vessels. So these histamines, they cause vasodilation, increase of the width of the blood vessel, and there will be phagocytosis by the WBC and macrophages, and these antigens or the pathogens will be eaten by the phagocytosis process. Okay, so inflammatory barrier, inflammation means swelling. If any antigen antibody interaction is there, there must be the swelling, and this swelling is due to the release of histamine from the mast cells, which cause vasodilation, increase of width of blood vessel. Okay, so that there will be more and more phagocytosis. So all these are the innate immunity, how it is maintained by 4 different types of barriers, and it is non-specific defense mechanism. It can kill any different type of microbes causing different diseases. It is non-specific. Then come to the acquired immunity, which is the specific defense mechanism of our body. Acquired immunity, from the name itself, acquired means it is gained, it is acquired either by medication or vaccination or by immunization. Okay, so first time Edward Jenner, he had first discovered and he had first applied the immunization process. The synthetic antibodies of a particular agent, a particular living organism, can be applied to the non-infectious living organisms, so that non-infected living organisms, so that there will be no further infection. And it is called immunization, which was first used by Edward Jenner, and he is known as the father of immunology. Immunology is the branch of science which deals with the immune system and development of immunity against diseases. Okay, so Edward Jenner, and this acquired immunity, it is the third line of defense. See, this acquired immunity, it maintains 4 characteristics. First character, 4 characters are there of acquired immunity. First one is specificity. This acquired immunity, it is specific for a particular microbe. Okay, means a specific type of antibiotic we take just to prevent the multiplication of a particular specific microbe causing a particular disease. And a particular vaccine, it prevents the disease formation and disease development in our body. Means this is a specific the specific disease can be avoided. Okay, so specificity is there. Then second is diversity. Large means certain antibiotics are there which are known as broad spectrum antibiotics. These antibiotics, they maintain diversity. Okay, diversity means different group of microorganisms causing different diseases are also avoided. Their multiplication is avoided and they are killed. This is called diversity. Diverse group of microorganisms, it can also act upon the diverse group of microorganisms, that is diversity. Third one is distinguishing the self and non-self. Whenever we are taking any of the antibiotics or taking any vaccine, then there should not be any interaction of these with our self cells, body cells. They should recognize which one is the self body cell and which one is the antigen or the foreign microbe. Okay, so this acquired immunity, it can distinguish the self cell and the non-self microbes. And fourth one is memory. See, this acquired immunity, if someone has taken the vaccine for a particular disease, then this particular disease causing microbe will not cause the disease more throughout the lifetime. Okay, this is the memory of the acquired immunity. The acquired immunity can memorize the entry of the particular pathogen into our body. Okay, so that there will be production of antibody against that particular disease. So there is specificity, diversity, distinction between the self and non-self, and memory. These are the four characteristics of acquired immunity. Then come to the acquired immunity. It is of two different types: active acquired immunity and passive acquired immunity. Active acquired immunity, it is gained by vaccination. By taking the attenuated, weak and or heat-killed microbes causing a particular disease, we can prevent the infection more throughout our life, and it is long-lasting, and there will be no reactions. Little amount of reactions are there in certain cases of vaccination. This is active acquired immunity. Directly we are taking the pathogen which is causing the disease. So it is the active acquired immunity, and it is long lasting. Then passive acquired immunity, for certain microbes, for certain pathogens, there will be no immune response in our body. There will be no antibody formation in our body. Okay, so for that we are taking synthetic antibodies or any of the antibodies which are produced in other animals, and these are injected into our body. This is called immunization. Synthetic antibodies are intaken, but these synthetic antibodies, they have a particular lifespan and they will be destroyed after a particular time. Okay, so antibody injection, it is there, it is called immunization, and it is short lasting, maybe also having certain side effects. Then humoral and cell mediated immunity, two different types of immunity are there. Humoral is the immunity maintained by the body fluids, blood and lymph, that is production of antibodies by the B lymphocytes. It causes agglutination and neutralization of the microbe. And that is the cell mediated immunity, already we have discussed in the previous classes. It is maintained by the T lymphocytes, and there are four different types of T lymphocytes: cytotoxic or killer T cells, which release porphyrin and cause pore formation in the wall of microbe, wall of pathogen, kill them. So these are killer cells. Second is helper T cell. It covers about 75% of the T lymphocytes. It helps for the production of antibodies. It also helps for the formation of more and more T lymphocytes. Then suppressor T cells, third one. The suppressor T cells, they are very active. These suppressor T cells, they suppress the whole immune system against the self cells. So it prevents the autoimmune diseases. The diseases caused by our immune system, these are prevented, these are suppressed by the suppressor T cells. And fourth one is memory T cell. These T cells, they memorize the entry of pathogen for the first time, and they can memorize it for a long time, and they are also ready for fighting against any of the infections. So T lymphocytes are of four different types, and they maintain the cell mediated immunity, whereas B lymphocytes, by producing antibodies, they maintain the humoral or body fluid immunity. Come to the lymphoid organs. Different lymphoid organs are there, distributed throughout our body, maintaining our immune system. So primary lymphoid organ, it is bone marrow and thymus. Bone marrow, it produces the B and T lymphocytes, and thymus, there will be maturation of the T lymphocytes. Then secondary lymphoid organs are there, like lymph nodes, spleen, tonsil, pairs present in the small intestine, and MALT. MALT is the mucosa associated lymphoid tissue. Mucosa associated lymphoid tissue, it is present throughout the mucosal layers, Then respiratory, digestive and urogenital tract, they also contain the lymphoid tissue. So they maintain the secondary lymphoid organs. These lymphoid organs, primary these are bone marrow and thymus, and secondary lymphoid organs are these one: lymph nodes and spleen, tonsil, patches of small intestine, malt, and in the respiratory, digestive and urogenital tract. These are the tracts through which any of the infectious agent can enter into our body. So these also act as the lymphoid. They are also layered by the mucosa layer, which maintains the immune system. Okay, so all these are the lymphoid organs. That come to the autoimmune diseases. Autoimmune diseases are caused by our immune system only when there is no detection of the self and non-self. Then some of the self cells, these are attacked by the immune system, causing the autoimmune disease, like Graves' disease. Whenever there is the thyroid cells, they are they are treated as antigens. Then overproduction of thyroxine hormone, it causes Graves' disease. Then diabetes mellitus, whenever the beta cells of isolates of Langerhans, these are attacked by the immune system, then there will be no more production of insulin, causing diabetes mellitus. Multiple sclerosis, it is the failure of muscle control. This multiple sclerosis, where the antigen means the immune system thinks the nerve cells and the coating of nerve cells edge the antigen, so that there will be no more muscle control. And myasthenia gravis, it is also a muscular weakness and fatigue disease. And here the antigen means the immune system, it treats the neuromuscular junction as the antigen, so that these different types of diseases are called autoimmune diseases, AIDS, and these are developed by our immune system by attack of the immune system to the body cells, to the own cells. Okay, so this is all about the overview of innate and acquired immunity. Innate immunity, the barriers, they maintain the innate immunity, and acquired immunity, it is maintained by either vaccination or immunization. Okay, and the humoral and cell mediated immunity also we have discussed. Thank you for watching. Share and subscribe my channel for the upcoming videos. Thank you.